Thank you, Sylvia. Good afternoon, everyone. I'm Jeremy Miller from the Understanding Evolution Research Group here at Naturalis. These days, my research is basically focused on two things: teaching computers to identify little brown bugs, which is something I am doing in collaboration with the ARISE project, and teaching computers to read taxonomic literature, which is something I have been doing for some time now in collaboration with PLASI, the organization out of Switzerland. My presentation today is about the latter topic. Specifically, I will be focused on linking material citations and taxonomic treatments. Material citations document the source of observations and data that appear in taxonomic publications. They allow us to go back to the original object. They are a key part of what makes the study of biodiversity a science, because examination of the original object can lead to reinterpretation or independent validation of the data. I particularly want to focus on the untapped value of these records to institutions and the potential of using bidirectional linking to enhance the value of collections data. But before we get into this, I want to take a minute to talk about taxonomic names, taxonomic treatments, and taxonomic catalogs, and how these all relate to each other and to the growth of knowledge about biodiversity. So whenever we use a taxonomic name, we are linking to information about one of more than 1 million species on Earth for which we have a name. This may mean that an individual of that species was observed at a particular place and time, or it may be something about the physical characteristics of that species, or how it interacts with other species in its environment. But if this information is associated with a number of different taxonomic names, all different in spelling, but pointing to the same real species in nature, how do we retrieve all the information known for a particular species? To help with this, we can turn to a taxonomic catalog. For example, I'm going to use the World Spider catalog to briefly take you through the taxonomic history of one species of spider. So here we see the accepted name, which is the name for this species as it is in current use. Next, we see the author and year of the original publication for this species name. The identifier is an LSID, or life science identifier. It's a machine-readable, unique identifier for this species. The treatment citations list all the published taxonomic literature containing content on this species. These point to a publication, such as a journal article or book, but more specifically, they point to a section within that publication that is focused on providing data about this particular species. This is what we call a taxonomic treatment. And this is our basic unit for building knowledge in biodiversity and taxonomy. But for now, let's focus on the example at hand. This species was originally described under the name Oresus famosus by Carl L. Koch in 1837. Then in 1865, his son Ludwig Koch described Oresus bubo as a new species. In 1878, Ludwig Koch published a redescription of Oresus bubo. Redescriptions are common in taxonomic research and typically summarize previously known information about a species while adding additional data. In keeping with standard scholarly practice, the previous description is cited in the redescription, so this treatment includes a treatment citation. In 1920, Tucker recognized that Oresus famosus and Oresus bubo were actually descriptions of the same species. So Oresus bubo became a junior synonym of Oresus famosus. Note that this means that specimens and observations recorded under the name Oresus bubo apply to the newly expanded concept of Oresus famosus. Here again are a series of treatment citations, including one where the synonymy of these two species is formalized. In 1967, Lehtinen established a new genus, Gondonamino, and placed this and other species within it. So this is a change in taxonomic nomenclature to accommodate new understanding about how this spider and its relatives are related to each other. Taken together, these taxonomic treatments contain practically everything that is known about this species. Taxonomic treatments include a taxonomic name, the identifier that allows us to accumulate knowledge about that species or higher taxon. Other common sections that may or may not be present include citations to previous treatments on this taxon, changes to taxonomic nomenclature, formal taxonomic description, diagnosis for distinguishing the taxon from all others, images depicting anatomical details, notes on biology and ecology, and citations of the specimens used to make the observations documented, which we refer to as material citations. Material citations are the link between the growth of biodiversity knowledge through taxonomic literature and the global network of institutional archives that preserve these specimens that this knowledge is built upon, which is to say, natural history collections like ours. So obviously spiders are a modest part of the entire tree of life. So what do we do if we are interested in knowledge on something other than spiders? Well, there's a global catalog of life, which happens to be based here at Naturalis. And this umbrella catalog aggregates many smaller catalogs, including the World Spider Catalog. But this means that the catalog of life is much more uneven in its completeness, especially when it comes to treatment citations and the different name forms that have been used to indicate particular species. So if we go to the catalog of life page for that same spider species, Gondonamina famosa, we find that the synonyms and treatment citations are missing. So now that you have an idea about what we mean by treatment citations, I'd like to present 3 vignettes involving linked material citations. So all of these studies tie in with an important collection here at Naturalis. The first of these involves damselflies in the family Lestidae. In my lab, we've applied semantic enhancement to a large portion of the legacy taxonomic literature of Lestidae, including the markup of material citations. These have all been shared with GBIF as part of the routine Plausi process. And we analyzed all records of Lestidae on GBIF, categorizing them by source as material citations from taxonomic literature, museum collection specimen databases, DNA databases, and human observations networks. The second study concerns Leocrenid spiders, performed by my collaborator and member of the audience there, Andreas. Hi, Andreas. Here we have highlighted the kinds of useful information that can be incorporated by museum collections databases via bidirectional linking. And finally, I will tell you about an ambitious project to mobilize all data and taxonomic treatments on one charismatic species. And since it's Naturalis, we have, of course, selected Tyrannosaurus rex. So we know, for example, exactly which specimens of T-Rex have been cited in which publications, as well as where they were found and where they are archived now. And we've documented inconsistencies in the specimen codes as they appear in the literature and highlight the importance of machine-readable unique identifiers to facilitate bidirectional linking. Our intention is to use the general appeal of this particular species as a hook for communicating broadly about biodiversity data and as a model for how biodiversity knowledge, born in taxonomic literature and supported by museum collections, can migrate into the educational space of museum exhibits and ultimately into popular culture. So, on to the project on lestid damselflies. So lestids are a mesodiverse family with about 150 species in 9 genera. They have a cosmopolitan distribution with concentrations in Southeast Asia and Australia. Furthermore, Naturalis has one of the most important collections of damselflies and dragonflies in the world. So for our project on the taxonomic literature of lestids, we've mobilized 224 treatments in 62 publications, and these cover 126 species. 61 of these have a total of 269 material citations records. So here we see Golden Gate Imagine. This is the Plausi developed editor for applying semantic enhancement to taxonomic publications. So note there we have this parsed attributes of this record, which ultimately allow this data to be mobilized. 150,000 GBIF records into these 4 categories: museum collections databases, human observation networks, DNA databases, and taxonomic literature. And this is a visual summary of what this looks like. So the size of the bubble is the number of species, and the position on the y-axis is the number of unique species, okay? So by far, the largest number of records comes from human observations networks. but these are especially dominated by common species. Museum collections records are more modest in size, but include a lot of species not found anywhere else. Taxonomic literature provides a comparatively small number of records, but these include some records of particularly high value, with a fair number of species not found elsewhere. So keep in mind that we semantically enhanced treatments on 126 species, which is nearly as many as the museum collections databases, but only about half of those species have treatments that contain material citations, which would be show up on GBIF. DNA is a growing component of biodiversity data, and in many cases, these data resemble material citations in the taxonomic literature in that they are derived from a specimen that can be returned to because it's archived in a natural history collection. However, a corpus of DNA data based on environmental sampling is emerging, and these are a bit more analogous to human observations networks in that they don't come from a specimen archived in a natural history collection. So we went through all of the records from taxonomic literature and DNA databases and looked for corresponding records in the museum collections data. So we were able to recognize about 15% of the literature records as having a match in a museum collections database. 29 of these were from our own institution, which we indicate here as RMNH. Nearly 40% of the DNA records are recognizable as having a museum collections record, and about four of these came from Naturalis. So here's one of our listed treatments in the Golden Gate editor here, and its appearance on Treatment Bank. So note we have this little green material view materials annotation in the Treatment Bank record. And this provides a link to that record on the Naturalis online collections database bioportal. So this link is created using this machine-readable persistent identifier that's provided by bioportal and has been added to the literature record. So this means that we're always going to know that these two records point at the same object. So through bidirectional linking, we can offer Naturalis the option to harvest this record as associated with their collection. So ultimately, we envision museum collections using such links to build metrics on the value that their collections are contributing to research. To touch on the DNA component for just a moment, and this is a feature still in progress, here are some elements from a publication featuring DNA sequences of lestids in the context of a phylogenetic analysis. So we can link a specimen to its museum collections record, its DNA sequence records, and its appearance on a phylogenetic tree. So what's next for lestids, and I think Donut will probably touch on this a bit in a few minutes, is to establish machine-readable links between the treatments through their citations of each other, in an effort to build an automated taxonomic catalog linked to the anatomical and biological content and the citations of the specimens and literature contained within. So that brings us to our second vignette, the one on Leocrenid spiders. So this started off with planning for fieldwork. And when you're planning for fieldwork, it's just sort of standard scholarly practice to gather as much information as possible about what is already known about what you're going and looking for. So in a paper published in 2020, we detailed a body of literature that we semantically enhanced with the intention of planning a collecting expedition. We extracted nearly 3,000 records from about 200 treatments across 200 publications. And because we mobilize these data through semantic enhancement, they are available for others to use and for other purposes. So here are two treatments of a particular Leocrenid species as they appear in Golden Gate. Here again are those parsed material citations records, which makes them mobile. And then they are tied through this persistent identifier to the corresponding record in the Naturalis collections database. And we can use, and we can see that with these, again, these little green view materials annotations. But here's another listed record from the literature that also contains some additional information that could be of value to collections. So this record asserts that this specimen in the Naturalis collection is actually misidentified, or at least has an alternative identification available. So through bidirectional linking mediated by a machine-readable identifier, collections could indicate that an alternative identification is available for this specimen, and that the provenance of these determinations is available on Treatment Bank. In addition, I want to highlight that there are data fields in the literature record that have a null value appearing on BioPortal, in this case, the collecting date. So through bidirectional linking, it would be possible to harvest such missing values. So finally, I want to go on to the Tyrannosaurus Rex treatment project. We now have mobilized 158 treatments in 105 publications on T-Rex, including its synonyms. These cite 165 unique specimens archived in 35 natural history collections. So we know where each of these specimens was discovered and where it's archived now. We also know which publications cite which specimens. So about 2/3 of the specimens cited are only in one publication, while a handful are cited around 30 times. Disambiguating all these specimen citations is not trivial. So here, for example, is the famous specimen Sue on display at the Field Museum in Chicago, and the various specimen codes that refer to it across different treatments. So this takes a little detective work. Of course, machine-readable identifiers make this all much simpler. So here is a literature record of our own T-Rex Trix in a publication by Anna Schulp and his collaborators, linked to our collections database. So here again, we have the potential to add content to the collections database, not simply the citation of the specimen, but field values, such as the missing locality data here. Of the 35 collections of specimens in this database, 4 provide a machine-readable unique identifier. Several others have records that we could link to online catalogs, but provide only a query-style link and not a persistent identifier. So as I understand it, query-style links frequently do not hold up over time. However, it may be possible to facilitate bidirectional linking through the use of GBIF stable identifiers in some cases. So I think we're all well aware that the acquisition of a T-Rex by Naturalis has been a major object for the promotion of public exhibitions. And we see this as an opportunity to examine the spectrum of biodiversity data uses, gathered in the realm of taxonomic research, supported and archived by natural history collections as objects for public exhibition and education, and ultimately as icons of popular culture. This series of images shows the first illustration of T-Rex in a taxonomic treatment, an early museum exhibit, and a movie poster, all depicting this upright kangaroo-like posture. Starting in the 1970s, biomechanical research revised our scientific understanding of T-Rex's posture, and this has been reflected in both contemporary museum exhibits like ours and in latter-day popular entertainment, such as this screenshot from Jurassic Park. Here's one of our 158 T-Rex treatments in the Golden Gate Imagine editor and also on Treatment Bank. We've provided treatment citations to link the relationship of treatments to each other with an eye toward building a dynamic taxonomic catalog where things like nomenclatural synonymy could percolate through all the associated biodiversity data. Here's a map of treatments in that project, a part of one anyway, with their links and relationships to each other, including synonymies, both proposed and rejected. And also as part of this project, we've mobilized more than 600 images from the taxonomic literature, figure citations. Here's just a sample, and we believe that this could be a resource for public outreach, education, and inspiration. So that's what I had to say on treatment citations and mobilizing data in taxonomic literature.